So we talked about, um, we started with the I, then V1, then all these V2s, V3s, and LOC and empty. So we pretty well concentrated this back end here for the last four lectures. Uh, Hopefully that dies away soon. Uh, what we're going to do today is the rest of the brain, all in one quick lecture. We'll particularly concentrate on this big blue area here called the parietal temporal occipital cortex, or PTO for short, and this big thing up here called the prefrontal cortex. So anatomically, the brain is separated into these four main anatomical regions. Back here, it's occipital, mostly involved in vision. This is the temporal lobe, and we found out already that this lower part here and the underside is involved in the what stream, which is involved in the where objects are coded, and we'll find out later that up here is where the auditory system lies. An important thing is structure here is the lateral sulcus, or the Sylvian fissure, sort of like a big, deep groove, so that this part is almost like a wing sticking out of the brain, like a something like a little leg that's separated from the rest of the brain. The other deep valley is this one here called the central sulcus, and it separates the parietal from the frontal. So those are the sort of only sort of two deep valleys that you have to sort of know about. And we found out that first information comes into V1, and that's back here. Well, it also comes into the primary areas for the other senses as well. One of these lies here, and that's where all your sense of touch comes from, and partly taste, and partly vestibular, but mostly touch. Your whole body is laid out here with a face here, and the leg is over the side here. Then over here, as I mentioned, in the temporal lobe, you have where your sense of hearing first enters the cortex. Now, we found out that besides V1, we have higher order visual areas, V1, V2, and V3. Well, similarly for these other senses, behind the area that the primary somatosensory cortex is, you have the secondary, and around the primary auditory cortex, you have the secondary. So it goes from primary to higher order, and after that, it pretty well enters all over these other areas, prefrontal and PTO, and within them, it depending on what the action is, or what the visual or auditory stimulus is, there's a network of feedback loops that get activated. They're dependent on what the problem is, and these association areas, that's where the different modalities combine. So hearing and touch and seeing. That's where attention lies. We'll see how attention gets shifted here. That's where planning occurs, and things are remembered. And in humans, this association areas form 80% of the cortex. And it's so in other species, like a small mouse or something like that, 80% is just involved in the primary sensory areas and primary motor areas. So in humans, this area is the thing that is expanded. Oh, I need to. So from the association areas, information then flows to the premotor areas, and then finally to the motor areas. Right here, this is where all your muscles get activated. And it's kind of neat that this is in front of the central sulcus, and the somatosensory areas was just behind the central sulcus. And all your body parts are laid out in sequence. So the structures involved in moving your mouth and saying things are located here. In front, in back, and behind here is all the structures that get information about your mouth and your face. So much what we do is you can think of it as a reflex. Some are simpler reflexes, like swatting a fly, and some are more complex, like writing down the name of an object that you just saw. And the complex tasks, like writing down the name, involve processing that goes through all these areas, okay? So from the primary sensory all the way to the primary motor. But something like swatting a fly, well, that's a very simple circuit from here to here. So what does the cortex look like? Well, we saw in the earlier lectures that the gray matter is the important part. And so that's more darker here. It's almost black in nature. This is all the gray matter. And what the cortex is trying to do is pack in as much gray matter as it possibly can in this sort of circle or cylinder. And to do that, it becomes folded. That's why you have all these folds to fit as much gray matter as possible. You could think of, let's say you had a paper bag and you blew it up, and the surface of the paper bag was your gray matter. Well, to get that into the smallest volume, simply crumple up the paper bag. That's basically what you're doing here. You're taking all the gray matter and shoving them into a small space as possible. Another advantage of doing that is that it makes the distance between some of the connections shorter as well. So this is where all your neurons lie. So this is where all your connections get formed, and where all your memories get formed. So you want as much of this as possible. And we have about 100 billion neurons. Now, to connect all these 100 billion neurons, we have these axons, and that's located in this what's called white matter. So this white stuff is where all the wiring occurs. So you can see Some wires will go from V1 to V2, some from V1 to, let's say, MT, and others across the corpus callosum to the other side. Having such these interconnections, it's amazing how long these wires are. If you took all these wires, you'd have several million miles of axons inside your tiny little head. Okay, imagine a string 7 million miles long packed in there, and so everything gets interconnected, and that has a problem because in some individuals, some area will start producing action potentials for no reason. And that activity then will quickly spread to other areas, and that will induce epilepsy. Okay. And it's also, so the connections then were memorialized, and we have each of these neurons connects to about 1,000 other neurons, and so we have something like 10 trillion connections in the brain. So you can imagine that at least 10 trillion things can be stored there. So the gray matter also requires more oxygen than other areas, than the white matter. And so what the brain does is flood areas that are doing the thinking with as much blood as they need, and for only the time that they need. And what's useful about that is that we can use the blood flow. If we measure the blood flow, we know what areas are thinking, what areas are being activated. And that's what functional magnetic resonance imaging does, or fMRI. So MRI gives you this structure, what's gray and what's white, but you have no idea what at the moment is being activated. But with fMRI, you can put any one of you in a machine and give you a task and see what parts of the brain are active at that moment. Now, this activity that you can see here, it lasts for several seconds, and so well, action potentials themselves occur in fractions of milliseconds. So fMRI is slow in measuring activity as compared to if you were able to record activity in the brain. But there have been several studies done in humans where before surgery they implant a little electrode and see what that area is doing. They measure the actual action potentials from that area. But fMRI is becoming a very important technique to be used. Almost everyone in this building is doing some sort of experiment using fMRI. And the machine that uses fMRI is sort of located between here and the UH. There's a sort of funny looking building that's attached to the Robarts that has several machines there that do all this measuring. They're huge magnets. You slowly go in a conveyor. You lie down, and the conveyor belt shoots you into the, slowly draws you into the magnet, and then you lie there for possibly half an hour, or an hour, or an hour and a half, and stay very still, because movement, of course, will affect the placement of your brain and smudge the signal. And people can, outside the room, watch what your brain is doing. It's something I had been working on for the last about 20 years, and it's fun. So, connectivity is a very important part of the processing speed of the human brain. Okay, each neuron connects to 1,000 other neurons, and they in turn connect to 1,000 others, and so forth. And so it's what distinguishes us from computers. A modern computer has what's called 64-bit processors. So they only have 64 connections in comparison. And that's a crucial difference. So 10 billion neurons, each with 1,000 connections, gives you 100 terabytes of connections. And most computers have a terabyte of disks attached to it, some 4, some 5, but we have 100 terabytes of connections. So no computer yet understands the question, like a rope is for pulling, not for pushing. Pushing, that's a concept that I don't think a computer yet understands, but a young child of 6, 5 years old really understands that concept. So, question. Suppose everyone in the world were on Twitter, and each one could tweet to 1,000 others. In how many tweets could you get a message out to everyone in the world? Is it 3 or less? Is it 4? Is it 4 to 10, or 10 or more? Let's say 3 or less. 4? Okay, let's hit the table. 3 or less? 4? 4 to 10. 10 or more. I think the largest sound was at 4, which was correct. Yeah, so if you multiply these together, in one tweet you reach 1,000, in 2 you reach 1,000,000, in 3 you reach 1,000,000,000, and in 4 you reach 1,000,000,000,000. And as far as I know, the world population is at almost 8 billion, so we're well over the limit. So in 4 tweets, you can send a message to the whole world. And the brain works the same way. So in 4 connections, a neuron can reach any other neuron in your brain. So messages can be sent really quickly. And this is an image done of all these wirings. So these are all pictures of the axons themselves going from place to place. And you can see how much, how many connections there are in the brain, how connected it is. This is where those millions of strings are located, going from place to place, and people are constructing what's called the human connectome, somewhat like the human genome. So what's a typical map of the human connections? What areas connect to what? So going back to the second lecture, you remember that you have gray matter, and it consists of 6 layers, 1 at the top, 6 at the bottom, and that information enters through layer 4, and it exits through layers 3 and 5. So we haven't seen that before. These 2 layers are where the output comes from. And in between, it has a lot of connections and makes a lot of decisions and stores a lot of memories. Now, layer 4, as we saw before, is that input layer. And it's thickest in areas that involve a lot of sensory information. So it was thickest in V1, and it's also very thick in areas that receive, let's say, all your touch information, your primary touch information. In contrast, this layer 3 and 5 are large in areas that send out a lot of output. And one of those areas is your motor cortex, because it sends information out to all your muscles. So you can see that different areas have different sizes of the different areas, different layers. And this fellow called Brodmann, about 100 years ago, by this criteria, by measuring where, what the thickness of different layers are, came to the conclusion that there are about 43 major areas in the brain. For example, if one looks at V1, that big layer 4 that you see there, as soon as you hit layer V2, that shrinks enormously, and you can see anatomically where V1 becomes V2, changes to V2. And every area of the brain, as we mentioned before, has columns. Now, how is the brain organized? What's the function of all these areas? Well, over the history of time, in terms of neuroscience, which is in less than 100 years, but Lashley, for example, was a fellow that came out with this equipotential theory. He thought that information is spread out over the same information. So let's say my memory of your face is not located in just one area, but it's spread out to the whole cortex. And so if I lose a small chunk of cortex, then that memory gets a little blurred, but not by much. Okay, still have the rest of it. Well, the evidence of that is that one evidence is that we do have different structures. We saw we have a V1. We saw we have a primary sensory area for touch, another one for the auditory system. So it isn't uniform. At the very other extreme, okay, is something called the grandmother theory. The idea is that, okay, you have V1 cells, and you have complex cells, then you have finally you get to something like the face cell, which stores grandma. Well, the, it, that's probably true. There is a cell that stores grandma, but there's lots of them. It's not just one, because you lose cells from day to day. About on average, a couple of thousand cells each day disappear for a variety of reasons, and you just don't wake up one day and forget grandma, but not grandpa. So there are many cells that store grandma, but they're all concentrated in this small area called the fusiform face area. Fusiform face area probably contains several million cells. So truth is somewhere between this. It's not one cell that stores grandma, but a bunch of cells, but they're not spread out all over the place. They're concentrated in small structures. The other thing is a particular area identical in everyone? Is your brain the same as everyone else's? Well, if a particular input is missing, let's say you had a cataract, and as a result of the cataract in one eye, you lost your binocular cells. Well, your cortex will be somewhat different from somebody else. Similarly, if one, rather than one input being missing, one area got damaged, well, another area could take over. For example, language, which is usually on the right, if at an early age that for some reason received damage, the other side can take over, and the other side becomes your language area. Also, the two halves of your brain aren't equal. Okay? Each side has its own specialization. In most, language is located on the left. And for that reason, it's called the dominant side. And it's called the dominant side because of neurosurgeons. Okay? You can easily sort of test a patient and find that he's lost language. It's an easy thing to measure one's language abilities. The abilities of the right cortex are more difficult to measure. And for that reason, the neurosurgeons claim that this area was dominant. But in fact, both areas are equally important. It's just that we haven't quite discovered what the important things of the right cortex are. We do know it's very important for things like faces, because the face area is larger on the right. And of course, language parts of the language has taken over parts of the face area on the left, because that's where other traits of language reside. The left side is also involved in things like reading, writing, signing, and it's also involved in problem solving, like sort of analytical, algebraic things like A equals B, B equals C, therefore A equals C. That type of logic. We'll also discover that this side is also involved in storing numbers. This side, on the other hand, the non-dominant side, is more involved in things involving parallel processing. So when you look at the face, you're looking at all the individual parts of the face at the same time. So, and you're deciding on the basis of all that information at the same time, whether that face belongs to this person or some other person. It's also involved in geometry, and geometry is, in a sense, similar because when you're comparing triangles, for example, whether these triangles are similar triangles, you're comparing all the angles at the same time, deciding, oh yeah, that one looks like that one's different from that one. And at the same time, things like whether C resembles O and I resembles L are important there, as is music. For some reason, the right side is more involved in music than the left. Okay, one of the interesting things that has been studied is people that have had their corpus callosums cut. Imagine cutting one half of the brain and separating it from the other. And that is it was done and is still sometimes done to relieve epilepsies. So if you've got a very severe case of epilepsy, if you cut parts of the corpus callosum, you limit the spread of the epileptic activity to one side. You don't let it spread to the other, and that causes the epilepsy to be much less severe. But what happens in patients that have their corpus callosum cut? Well, let's suppose the person was looking at something like this, an apple on this side. Okay? That apple is projected to this side of the brain. Okay? So this side of the brain sees an apple, but the person, because the language is over here, can't recognize and say the word apple. But the left arm is represented here as well. So the left arm can reach for this apple. So this is sort of the difficulty that a patient like that would have. The other thing to think about is that if you have a patient like that, and one side is separated from the other, you in a sense imagine having two of you in one head. And one side had no idea what the other side was thinking or experiencing. It's a weird thing to imagine, two of you in one head. And there's examples of patients that are thinking very different things. One patient, for example, would hug his wife with one hand and push her away with the other. So that's one example. Now, why do we have lateralization? Why is one side specialized in doing one thing and not another? Well, one advantage is that the corpus callosum is a long pathway to go from one side to the other side. It's easier, if you want to do one function, to concentrate it in one side of the brain. so you have shorter connection. The other thing is that having two sides do the same thing is sort of a waste. It's better to have one side do one problem and the other side do another problem. You have twice as much capability if you do it that way. The disadvantage is that there's less redundancy. So if one side gets damaged, that area loses that function. And of course, if you have damage of the language area as an adult, you will not regain your ability to speak. Okay, now let's look at the function in detail of these two association areas. The first one we'll look at is this prefrontal area. That's really expanded in size. You can see here how it's expanded over different species. And it appears that this is what distinguishes us from other animals. Now, the gorillas are bigger than humans, but their brains aren't bigger than ours. And the question is, why? Well, the problem with having a big brain is it takes up a lot of calories. And gorillas haven't invented cooking yet. The thing that distinguishes us from most all other animals is that we can cook. Being able to cook is a huge advantage. Our digestive system can work can pour out, digest the calories in food much quicker if it's cooked. And as I mentioned many times, thinking is expensive. The brain, which is only 2% of your body weight, okay, consumes 20% of its oxygen and metabolic consumption. So things all like calories. So without cooking, to sort of feed a brain our size, you'd have to be eating all day. You have no time for thinking or going to class or anything like that. Constant looking for food and eating it. So let's test your prefrontal cortex, okay? One of the things that the prefrontal cortex is involved is spatial working memory. By working memory, I mean things like on a chalkboard, okay? You write something down, and then you can erase it and write something else down. So you're thinking about one thing, and then you switch to another thing. And it's what's sort of a temporary storage. So this is one thing that the prefrontal cortex is good at. It's temporary storage of facts all around you. The other thing about it is that often the type of working memory it's using is spatial. So is that something over here, or is that something over here? So to test it, I want you to remember where this egg is. Okay. Okay. Is the egg under here, or is it under here? How many people think it's over here? How many people think it's over here? Oh, the class was right, or the ones that offered a sort of weak bang. Okay, so if you have a lesion of prefrontal cortex, you won't have this ability. Interesting enough, children before the age of 1 haven't quite developed their prefrontal cortex. So when toys are out of sight that are mind, you can put a cover over a toy, or let's say a cover over two covers, one over the toy and one not over the toy. The child will have difficulty remembering where that toy is, or that a toy was there. The other thing that's different about it is that you saw that an egg appear here, and that set off V1 right away, but then the egg went off and V1 became silent. In contrast, this prefrontal cortex kept going. Also, an egg appeared here, and neither of these areas were active. So this is the fact that only an egg on one side get activated for a long time, that's an indication of working memory, and the fact that it's only on one side and not the other side is an example of its spatial abilities. So different cells and code different locations. Now, dopamine is important in fueling this working memory. And that prefrontal cortex is interconnected with a structure called the basal ganglia. If you have some deficit in the basal ganglion, you develop something called Parkinsonism. And Parkinson patients have, for example, difficulties in initiating movements because they don't have a target for their movements. Okay, there's a good example in a movie that I saw about Parkinson patients. They took a bunch of Parkinson's patients are usually very catatonic. You can place them where you want and they won't move. And so the doctor there placed them all in a row, like 5 in a row facing one way and 5 in front of them facing the other way. And then, so they were doing nothing. Then he tossed the ball into the middle of them. And just the sight of the ball allowed them to pass it back and forth between each other. So one row would pass it to the other row. So they can't initiate a movement because they can't sort of create a target in their working memory, but they can respond to one that appears there, but not for long. So having this ability for working memory is very important in making decisions and planning. You can imagine looking at a chessboard. You have to remember all these places of where the chess pieces are in planning your moves. But after lesions, they found that patients became calmer and less frustrated. And that calmness was for an important reason. If you can't plan something, well, one doesn't have any expectations as to what might happen, and therefore you don't get frustrated if things go wrong. If you don't have a plan, there's no expectation that it's going to succeed. So because of this calming effect, something called frontal lobotomies became popular. So they would make a cut here, separating this part from the rest of the brain. And people, of course, that would cure things like aggression. Unfortunately, it also destroyed initiative. A fellow by the name of Moniz, a Spanish fellow, won a Nobel Prize for coming up with this technique. So, you know, I think most of us would be kind of shocked that someone would get a Nobel Prize for something like this. And in the U.S., a doctor called Walter Friedman used an ice pick that he would drive through the back of the eye socket and then twist it back and forth, and he would then use that to separate the frontal lobe from the rest of the brain. And he would do that in minutes in his office. And this is an example of him doing that to a patient. And these are all would-be doctors looking at this technique with interest to be able to do it themselves. And it wasn't, you know, like people without much thought that had this done to them. One of the patients was Rosemary Kennedy, who was a sister of John Kennedy. And of course, after this, she became largely a vegetable. So, one of the reasons I bring this up is this, you know, a Nobel Prize winner coming up with this technique and it being used on many people. That to us now looks very unacceptable. And if many of you become doctors, I'm sure that be careful that some of the techniques that are taught to you in 20 years later, you say to yourself, oh, that was awful. Okay? Keep a lookout. Beware. Okay, so we'll move on now to this structure back here. It's a big one. It involves the parietal, temporal, and occipital cortex, a little bit back here in the occipital areas. And it does a lot of things. One of these is polymodal convergence. It's a big word for just sort of locating objects in space and combining things by touch, sight, and sound. So you can imagine that a structure here is in the middle of a touch area, a hearing area, and a visual area. And so it's strategically located to be able to interconnect all these areas. And that's what this convergence talks about. It's also involved in attention. It allows us to focus in on a specific stimulus and ignore others. Like if you're in a crowd, you can focus on a particular person and all the other noise disappears. And it's also where memory lies. And that's especially true for this part underneath here, the inferior and medial part of the temporal lobe. And we won't talk much about this today, but we'll talk about this in the very last lecture, lecture 12 of this course. So, attention. This fellow here is on a motorcycle and he's wearing sort of a clown's outfit. And he's riding around something like the UCC square there outside the UCC. And students are then, that sort of pass by this guy, are asked, did you see anything strange? And half the people carrying cell phones said they did not. They walked right by this fellow and didn't notice him. Okay. So where your attention is focused can make you blind to huge things that are happening around you. And that's true whether you're walking or driving a car or whether you're listening to this lecture. Depends what you have on your screens at the moment. Okay, to give you an example of that, can you spot what is changing in this picture? Can you see something change? Raise your hands if you spot something. Don't say what it is, but slowly, 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 more and more. Okay. The bicycle is disappearing. No. Okay. So if I circle it, then you can see it clearly. The bicycle is appearing and disappearing. So see how that circle now has focused your attention to this small spot. But when your attention wasn't focused to that small spot, you didn't see it. And there's on the links to other neat things, there's examples of other pictures like that you just don't see huge things like the engines on an airplane appearing and disappearing. So one analogy of attention is like a flashlight. It focuses your attention to one spot or another spot, or one feature or another feature. But attention is also like a bottleneck. Once attention is focused on this particular spot, it takes time for the information to enter and register in the brain. And while it's doing that, you're blind to everything else. One theory for why it's doing that is that if every area in the brain were active or admitting this information, then it's too much for the brain to handle, and the brain would fry. Well, we know that's not true. We know that instead of frying, it would simply faint. So it takes a lot of metabolic activity to activate brain cells. And that's one way, by using attention, you limit the activation. So here you can see that attention is this flashlight, and your attention is drawn to A, and it takes time for this A to be passed on to higher areas. And while it's being passed, you can see that if another ray appears or another object appears, it's blind to that object, even in this area. If another object over here appears, it's also blind to it. So it's like a bottleneck to processing. Now, attention can be drawn by something unique, like these lines are different from these lines. And so your attention is focused at these lines, and attention accentuates them and then lets that information enter the higher areas. Or you can decide what you want to look at. Here you decided you want to look over here, and A enters your cortex. Then you want to look at B, and B enters the cortex. And now you want to look at C, and C enters your cortex. So you can drive these things by uh, your will or external functions. Here you can see easily that it's where the event is happening. The bicycle is disappearing, and the reason that's that you can easily see that is that just the flash of it draws your attention to it, and you register it. In the previous one, what I tricked you with was having this blank, okay, a blank frame, and that blank frame disrupted, everything changed, and as a consequence, you couldn't tell that it was just this thing that's changing. Here, we can see that it takes time to find Waldo. Okay, and that's because Waldo doesn't pop out. Okay, there are a lot of things that resemble Waldo in this picture, and it takes time to find him over here. Okay, and it takes time because you don't automatically draw your attention to it, and because shifting your attention, you shift your attention to different places, and each shift takes time. And you can shift your attention either covertly, without moving your eyes. So you just look at one person and shift your attention to somebody else while looking at that same person. Or you can do it overtly by making an eye movement to it. But both those functions takes time. And then it takes time when you're either overtly or covertly attending to it to decide, is that Waldo? So if not, then you have to look at something else. Attention is not the same thing as arousal. Arousal, something like drinking coffee or something like that, accentuates your arousal. And that's mediated by structures like this one here from locus coeruleus. And that releases something called norepinephrine. And that sort of tunes up the volume of all your cells, okay? It's like a volume control on everything in your brain. But it's not selective to a particular region or a particular function. Just increases your overall alertness. Okay, let's look at an example of attention. What I want to do here is you're looking for the circle, okay? The circle here. And how long does it take you to find the circle? Well, you might have noticed that a square appeared here just before the circle appeared. That's a cue for where to look for the circle. So it draws your attention. So if you could hit the table as hard as you can when you see the circle. Okay, let's try that again. Hit the table a little harder. Not bad. I tried to trick you by drawing your attention to the opposite side, but you were pretty good. That was a bit slower because it came up in an unexpected place. So you can see how your attention can be drawn to a particular location. and your reaction can be slowed by that attention being drawn to that location. Then your attention can be, different areas can be involved in attention. So here, if you are asked to click if two objects are of the same color, so I'll start it up, and so that you were supposed to bang your hands at that point. Dunk, dunk. And if you were doing that, I'm not sure I got this programmed right, but if you did that, then maybe this area here involved in color processing would become active. If instead you were asked to look at shape, if two objects are the same shape, again, that one would be the right one to do. LOC would become active. Okay? And finally, if we asked you to click when two objects are moving in the same direction, okay. So again, oops. In that case, if I click right, MT would become active. So you can see that as the function changes, different areas get activated and involved in the task. And so, again, not every part of the brain is being activated. Again, you're saving oxygen and concentrating your activity to a particular thing. Another task. Can you see what's different here? Can you spot the difference? Well, of course, it's this line here, right? Can you spot the difference? Oops, back. There you go. Now, in this next one, can you spot the difference? I did it yesterday. Okay. Can you, it's this thing here, right? But it takes a while. Wow, it's more difficult. So you have to, again, look through many more areas. Now try this, okay? Try to sort of attend to the black lines, okay, in the next one, okay? Sort of, you know what it's going to be, the black lines, and your brain can filter out the white lines, and you can see it right away. Which one? This one here. Okay? Focusing on a certain color will allow you to attend to a certain object. So you can see how focusing the different attributes, focusing different parts of your brain to the task can speed up your decision processing. And of course, that's using different parts of the brain. So we saw the two types of attention. One was spatial, where you voluntarily directed things to the left or right position. I got that square to appear here, 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 and you attended that spatial location. Or it's featured based. It depended on, let's say, a color, a shape, so the brain can attend to that quality. In addition, there's a third type of it can be object-based. So here, an object appears. You can clearly see it. Here, a line stands out. Here, you can see a letter stand out. So these are different objects, and they stand out from other objects. Now, we talked about attention. The opposite of attention is neglect. If you have a lesion of the right parietal cortex, you get neglect. And you get neglect of the object's left side. So a patient, if you had this lesion, you would be unaware of the other side, even though you're making a portrait of yourself. And again, in the links to unique new things is that there's an example of an artist who had such a lesion, and he made a portrait of himself, and you can see that portrait. It's missing half his face. But gradually over time, that patient recovered. And you can see as over the months, the other side appearing in his portraits of himself. Now, having I call this a PTO lesion. I should really call it a parietal lesion, but PTO encompasses the parietal. So if you have a lesion, you can see that it's different from a V1 lesion. If you have a V1 lesion on one side of the brain, if the object is here, you can see it. If the object is on the other side, you can't see it. Here you're missing the left side of the face, no matter where it's appearing. It's because attention grabs the object, and it sort of makes like if you imagine a square, a frame around the object, and it's the object itself, and within this frame, that is the left and the right of what you can see and can't see. Now, you might imagine that a lesion of the opposite side would have a comparable effect. But for some reason, that's not the case. And one theory is that on the right, you have representations in the parietal cortex of both this side and this side. Whereas on the left, you only have this side. So when you have a lesion on the right, you get rid of the left and the right, and you miss out on everything over the left. But since you've got a right over here, you don't miss out on things on this side. In contrast, when you have a lesion on this side, you can see both sides because it's still being represented on your right parietal cortex. So there are many ways of stating an object location. We'll look at coordinate frames. So here we see an object on a table. So our reference point, let's say our attention is focused on this table, and this object is located within this table. And I've drawn this ruler on both sides. So you're measuring the object's location with respect to what you're attending to. In this case, it's the table. So this is called an allocentric coordinates. Another name for it is object-centered, because you can think of the object being the table, and that's your frame of reference. Now here you have what's called an egocentric coordinate. In that egocentric coordinates, the rulers are you. Okay? Here's the ruler, and it's centered about where you are. But that you will see could be many things. It could be your body, your head, or your eye. And we'll see in next week's lecture, or the lecture in 2 weeks' time, how this can occur. Now, to grasp what's different about these two ways of thinking about allocentric or egocentric, one way is to make it two transparencies. The yellow one is an egocentric one, representing you in the center. The allocentric one is this one in gray, and it represents, let's say, a table with two objects located on it. Here you can see you moving in an allocentric frame. Okay, so it's the distance of you to these various objects that's being measured, and when you move, these objects stay still within this frame, but your position changes. In contrast, when you're thinking of things in the egocentric way, it's you that stays constant, and these objects move, so their location shifts. The other thing you could have is nested frames. So you can see, imagine this is a room, this is a table, and this is an object on a table. And you can imagine the object is the apple is being measured with respect to the table, and the table is being measured with respect to the room. Now, what's interesting about this is that these two different frames of references are represented by these two pathways that we've been studying. The ventral stream treats things in an allocentric or object-centered reference frame, and that's what you might expect, because you think about things like faces and features within faces, so it's a good way of representing things. This one up here, you remember, is the pathway that's involved with locating where your mouse is with respect to you. Okay? And a good way of expressing that is in an egocentric frame. You want to reach for it, you want to know where that mouse is with respect to you. You don't want to calculate it with respect to the table, and then where the mouse is with respect to the table, and do all those calculations. You just care where it is with respect to you. Okay, so imagine that if you're standing in the square and you want to walk to a restaurant, the where stream will be involved in coding the location of the restaurant with respect to you in an egocentric frame. In contrast, when you want to recognize a face, the what stream is involved in telling you what the details are of the face, of the features of the face with respect to the face itself. And that's it.